Ladies and gentlemen, it's my great honor to present my study entitled Increased Sensitivity of Central Serotonergic Neurons to Corticosteroids Following Maternal Immune Activation, Significance for Mood Disorders. This work was performed by my colleagues Matej Rachitsky, Daniil Grinchi, Roman Degtyarenko, Ruslan Polyoha, Yana Gaburyakova, and Marta Gaburyakova, and myself, in the Institute of Molecular Physiology and Genetics of the Center of Biosciences of Slovak Academy of Sciences in Bratislava, Slovak Republic. These are my disclosures. What is the background of our study? It is known from previous epidemiological studies that acute infective illness during the pregnancy may increase the risk of the development of psychopathology such as depression, schizophrenia, and autism spectrum disorders in offspring. To investigate the effect of maternal infection of offspring, there is a modern model which can be used. There is a model called maternal immune activation of MIA by lipopolysaccharide. What is liposaccharide? It's a component of the cell wall of gram-negative bacteria. This molecule is detected by cells of the immune system, and activation of these specific receptors, TLR4-MD2, results in increased expression of inflammatory cytokines. These cytokines like interleukin 1A, 1, 6, 8, or 12, or tumor necrosis factor alpha, they can pass the blood-brain barrier and affect the development of the embryo, including the embryonal brain. It was previously detected that maternal immune activation has depression like has anxiogenic and depressogenic effect on the offspring. What we can see here, that maternal treatment with lipopolysaccharide decrease the time spent by offspring rodents in the open field of the elevator plus maze apparatus. And what is very interesting, the effect get more strong of LPS when it's applied in the last trimester of the pregnancy. And this effect get even strong when offspring become adult rather than in juvenile offspring. What is known about neuroendocrine effect of the maternal treatment with LPS? It does not affect the corticosteroid levels in the basal corticosteroid levels, but it's increased corticosteroid response to the acute stressor applied on offspring in the later life. Acute stressors such as maternal isolation, restraint, or restraint. In our previous study, we tested the hypothesis that the behavioral effect of the maternal immune activation might be mediated via serotonergic, noradrenergic, or dopaminergic pathways. And we did involve that both in male and female offspring, maternal treatment with LPS during the gestation decrease the excitability of serotonergic neurons, which may explain the anxiogenic effect. But what about dopamine system? Without increase in dopamine neuronal firing activity, but this was found only in the male offspring. What are possible mechanisms of these neurophysiological findings? It is known from previous ex vivo studies in brain slices that the maternal treatment with lipopolysaccharide decrease excitatory postsynaptic currents in serotonergic neurons in the sorry, that corticosterone, application of corticosterone to the bath solution decrease the excitatory postsynaptic currents in the dopaminergic, in serotonergic neurons in brain slices. It may explain that the increase in corticosteroid signaling by MIA may decrease basal serotonergic neuronal firing activity. And what about this increasing effect on dopamines? It is known from our previous studies that, that serotonin decrease the firing activity of serotonergic of dopaminergic neurons of the VTA, and this effect is mediated by 5-HT2C receptor. It means what may happen, MIA, via corticosteroid mediated mechanism, may decrease the firing of 5-HT neurons. We have less serotonin, and because we have less serotonin, dopaminergic get activated. But this mechanism is found only in males, probably because males, they have stronger serotonin dopamine interaction. And in the present study, we wanted actually to test this hypothesis. First, to examine the effect of the maternal immune activation on the sensitivity of 5-HT neurons of the DRN. And second, to assess the effect of MIA on 5-HT dopamine interaction in male and female offspring. The methods were like used in our previous studies. The animals were allowed, male and female rats were allowed to mate. From females, we took daily vaginal smears, and when we detected the sperm cells in female vagina, we considered the day 0 of the gestation. During the days 15 to 19 of the gestation, we applied LPS in the rising doses from 20 to 80 microgram per kilogram, via rising doses to make more appropriate model of infection, because during the development of infection, also the immune response getting stronger. On day 21, we have usually birth happens, then animals were left untaken, allowed to grow up till the adulthood. And on the day 48 to 56 of their life, we performed in vivo electrophysiological investigations. In vivo electrophysiology, we anesthetized animals with chlorophyll drug and put into stereotaxic frame. Then we inserted glass electrodes filled with electrolyte to the dorsal raphe and ventral tegmental area. And we identified serotonergic and dopaminergic neurons according to their wave of the action potential and pattern of the generation described in our previous studies. In order to assess the effect of glucocorticoid on serotonergic firing activity, we used dexamethasone cyclodextrin complex, which is very fast past blood-brain barrier. And we administered this compound in the rising doses from 1 to 5 milligram per kilogram during recording from a specific serotonergic neurons. With respect to dopaminergic neurons, we applied first an agonist of 5-HT2C receptors and detected decrease in the firing activity. Then we applied an antagonist, and this inhibition was usually recovered. This is our result. We found that in both male and female rats, dexamethasone dose-dependently decrease the firing activity of serotonergic neurons. It was very interesting observation. We more or less confirmed the finding of the previous ex vivo study, now in vivo condition, and we indeed find that glucocorticoids inhibit serotonergic neurons. And what also we found that maternal immune activation increases sensitivity of serotonergic neurons to corticosteroids. With respect to dopaminergic neurons, the findings were even more interested. In female rats, in control, in maternal immune activation groups, we found dose-dependent decrease of dopaminergic neuronal firing activity induced by an agonist of 5-HT2C receptors. What about males? In control male, we found also the same decrease, but effect in MIA males, male offspring, was extremely interested. We found not only decrease, not only one decrease, but found even paradoxal activation of dopaminergic neurons by a selective agonist of 5-HT2C receptors. What our conclusions? That the activation of glucocorticoid receptors in vivo condition leads to the suppression of firing of 5-HT neurons, as I was previously observed ex vivo. It means that maternal immune activation enhances inhibitory effect on 5G neurons by the mechanism, probably using glucocorticoid, that it was hypothesized in our previous study. Decreased spontaneous tonic activity of 5HT neurons in rats is likely to be, as I said, it's likely to be resultant from increased sensitivity to glucocorticoids. Enhanced inhibition of 5HT neurotransmission for glucocorticoid might be responsible, at least in part, for the anxiety and depressive-like behavior characteristic in the offspring of rats experienced maternal immune activation prenatally. Activation of 5HT2C receptors leads to the inhibition in control animals, to the inhibition of 5HT neurons. And this is in line with our previous findings that antagonists of these receptors activate dopaminergic neurons, and maternal immune activation paradoxically reverse the inhibitory effect of 5HT2 receptor, and this might be responsible for increased firing activity of dopaminergic neurons observed in male offspring of the MIA rats. And alteration within the functional interactions between corticosteroids and monoamines might be of critical importance in pathophysiological mental disorders triggered by maternal stress and infection. I would like to thank the organizations which funded this research. It's a Slovak Research and Development Agency and the Research Grant Agency of the Ministry of Education of Slovak Republic and Slovak Academy of Sciences. You can listen and download this talk using this QR code on the left, and using the right QR code, you can see and download the poster on this topic, and poster is also available in the poster area. Thank you very much for your attention.